Welcome back to the channel Bio Academy. Myself Dr. Vasudha. Today we are going to study about paper second, section 5, Developmental Biology, in this subsection, that is P, Gametogenesis, fertilization and early development, human gamete production, along with the 7th section, that is System Physiology, animal. It includes endocrinology and the reproductive process, that is hormone, which are involved in this process we are going to study. Before moving to the entire structure and function of the human reproduction process, let's study some of the important events, gene involved, and important hormones and its regulation during the early embryo stage to the puberty. As we all know, the sex organ development and the sex differentiation takes place in the embryonic stage only, in mother womb. As we all know, in most mammals, sexual differentiation relies on the Y chromosome. Okay? There is a pair of endocrine structure that is known as testes in male and ovaries in female. Then, what is gonads? Gonads are reproductive organs in animals, including humans, that have two primary functions. One is gametogenesis, that is production of gametes. Second one is the production of hormones, production of sex hormones. Male gonad is called as testes. They produce gametes called as sperm, that is spermatozoa. In the female gonads is the ovary. They produce egg or ova. The hormone is involved in the male is the testosterone or androgen, and female sex hormone is the estrogen and progesterone. Okay, both hormones typically present in both sexes. Ovaries predominantly secretes estrogen and small amount of androgen also, while this pattern reverse in male. In male, what happened? The huge amount of testosterone or the androgen is produced and small amount of estrogen and progesterone are produced. Progesterone is a steroidal hormone which has impact on or important role in the pregnancy or preparation of the uterus for the pregnancy. Okay. Then next, moving to the next topic, that is the sex differentiation and development. The sex differentiation and development. The sex chromosomes. The sex of the individual is determined by the two chromosomes called as sex chromosome, which distinguish from the other chromosome, that is somatic chromosome, also known as the autosomes. In mammals, including human, sex chromosomes are called as X and Y. Male diploid cell contains one pair of sex hormone, that is X, Y, along with the somatic chromosomes. In female, diploid cell contains pair of sex hormone, that is X and X, along with the somatic chromosome or autosomes. So, Y chromosome is very necessary and it plays an important role in development of male sex characteristics and formation of testes. In Y chromosome, a specific gene is called as SRY gene, also known as sex determining region of Y chromosome. This gene is code for DNA binding protein. This DNA binding protein, which helps in the transcription, which act as a transcription factor by binding or binds to the DNA and bends the DNA and initiates a cascade of genes necessary for testicular differentiation. and development of the testes. For example, one of this gene is produced by this, that is from sex-determining region of Y chromosome, is the Mullerian inhibitory substances or MIS, which helps suppress the development of female reproductive structure in male. So it is very essential for the male development of male characteristic and prevention of development of female characters in male. So usually near the tip of the short arm of Y chromosome. If we consider the gametogenesis and genetic inheritance, during the formation of gametes, that is for sperm or the egg, through meiosis, every normal egg or the gamete contains X chromosome, but female diploid cell contains XX chromosome, but the gamete's egg or a sperm contains single sex chromosome either. In female, it in be X only, but in male either X or a Y chromosome. But in case of sperm, it is X or Y. Okay, half of normal sex carry a X chromosome and other half a Y chromosome, which is 50-50 split. If fertilization occurs when the egg usually contains X chromosome, when the sperm is having X chromosome, then fertilization happen. The pattern, the product pattern will be the XX, that is female pattern. Female is the product. If the fertilization occurs between X and Y chromosome of the sperm, then the pattern leading to the production of X and Y, and it is the male pattern. Okay, understood. Then let's moving to the human chromosome. If we consider human chromosome somatic cell which contains 46 chromosome, that is in male 22 pair of, that is 44 chromosome of somatic chromosome, 22 pair, and 1 pair of sex chromosome, that is X and Y. In female, it is 22 pair or 44 chromosome of somatic chromosome or the autosomes and X and X chromosome. Okay. In human or mainly in the female, there is a important event happen. That is X chromosome inactivation in female bodies. Okay. During the embryo stage only, the sex chromosome X hormones are produced, and the formation of gonads or primitive gonads are all produced in embryonic stage only. So, during the embryonic development, a remarkable process happens where one of the two X chromosome in the somatic cell of normal female became functionally inactive. One of the X chromosome of the somatic cell in the female cell that become inactive. Another one is the active. Okay. Why this is happen? This happens to balance between sexes. As we all know, mammals including human, female typically have 2 X chromosomes, while male have 1 X and 1 Y chromosome. Then, if female have double the dose of X chromosome genes compared to the male, if the balancing mechanism absent, it could lead to a imbalance in the gene expression. So, X-linked genes expression is more in female compared to male. So, we should have one balance between these X chromosome gene expression, so that the nature only selects and nature only do this for balancing So one of the X chromosome of the female somatic cell become inactive. That means in female somatic cell gene, one of the X chromosome are effectively silenced and does not show any expression. So here both male and female have an equal dose of X-linked gene products. Okay? Is that clear? Okay. This process is essential for normal development of female also. Without this process, the double dose of X-linked gene could lead to the overexpression of certain genes, so that it potentially causing developmental abnormalities in female also. And also some of the other health issues also occur. So this silencing of this X chromosome is very essential for the normal development of female. So it is very essential for the normal development of female also. So the choice of X chromosome in inactivation process is very random. It's a random process, and there is this process happens every single cell division, every single meiotic cell division, every single mitotic cell division. This silencing of X chromosome is happen, and also it's strictly a random process. There is no rules and there is no such criteria to inactivate the X chromosome. So this inactivated X chromosome is known as Barr bodies. That is, condensed and inactivated X chromosome is present in the somatic cell is known as Barr body. So each somatic cell of female body is containing typically one Barr body, which is representing the inactive X chromosome. One active X chromosome and one Barr body is present in every single female somatic cell. Okay, is that is clear? Then let's moving to other topic, that is X chromosome inactivation center and CTCF. Okay, the X chromosome inactivation center is present. Where it present? The initiation of X chromosome inactivation is orchestrated by the X inactivation center in the chromosome. The factor involved in this process is known as the CTCF or the CCC-TC binding factor. The factor which is involved for the inactivation. Okay. Consequent of this inactivation in adult female, some somatic cell contains active X chromosome which is come from the father or the paternal origin, and some are contains an active X chromosome of maternal origin. So, okay. Is that clear? Is that clear? Okay. So it's a random process. So some cell contains X chromosome from the father, and other cells may contain X chromosome from the mother. Another one important distinct feature of this process is the presence of small drumstick of chromatin, which is projected from the nuclei of a subset of polymorph nuclear leukocyte or the white blood cell in female. This characteristic is not present in the male. So this characteristic which distinguish the male and the female, that is the drumstick-like structure which is projecting from the nuclei of a subset of the polymorph nuclear leukocyte or the white blood cell in the female. Okay, is that clear? Okay, we discussed about the chromosome types in humans, sex chromosomes, inactivation of X chromosome, and why it is happen, and what is the benefit of this process. There's some other terminologies, that is the Barr bodies. Barr bodies are important. they are condensed and inactive X chromosome, as well as the other terminology, that is X chromosome inactivation center, CTCF, or the CCTC binding factor, which was involved in the inactivation of X chromosome. And mainly the X chromosome inactivation is a random process. Next, we study or discuss about the embryology of human reproductive system. Okay, in embryo stage, how the gonads are developed in both sexes we discussed here. First is the development of gonad. So let's dwell with the functioning process of development of human gonad, which are the process that is gonad is the precursor of the testes in male and the ovary in the female. Gonad is the primitive structure or the precursor for the sex organs in both sexes. Okay. During early embryonic development, gonad structure is quite similar in both the sexes until they approaches the 6th week of development, and the structure of gonads are essentially identical in both male and female. Then when differentiation starts, for genetic male, that is XY, the process differentiation begins during the 7th and the 8th weeks of development. In the embryo, a primitive gonads arises from the genital ridge, which is present in the primitive embryo, which is condensed tissue near the adrenal gland. Okay, in embryo, there is a genital ridge is present. Near this ridge forms a condensed tissue. That condensed tissue which is present near the adrenal gland. Okay, this primitive gonad develops a two structure, that is the cortex and the medulla. Until 6th week of development, the structures are identical in both sexes. Okay, cortex and medulla. This is the primitive structure or the pre-gonadal structure in both the sexes. Then in males, what happened? The medulla portion, the medulla region, developed during 7th and 8th week into a testes, and the cortex is present that is degenerate and shrinks and loses its potential and its function. Okay, only the medulla portion which is developed into sex. This is due to the genes and hormones, including expression of the SRY genes, which is present in Y chromosome. Okay, and secretes a testosterone as well as Mullerian inhibitory substances by these developing testes. The circular cell in the testes, which secretes the Mullerian inhibitory substances, also known as Mullerian regression factor. Okay, this Mullerian inhibitory substance is essential for the suppression of female sex character. This testosterone which is produced in the developing testes, which metabolites to form a dihydrotestosterone, which induces the formation of male external genitalia. This hormone is very essential, that is active form of testosterone is known as the dihydrotestosterone, is very essential, plays a very important role in the external genitalia and the secondary sexual characteristics in the male. This Mullerian inhibitory substance continues to be secreted by the circular cell of testes, and in boys, this Mullerian inhibitory substance level in plasma reaches peak of about 1 to 2 years of age. Subsequently, this Mullerian inhibitory substance level decline and low level by time of puberty, but it remains detectable throughout the life of the male. In the genetic female, what happened? The story is different. Okay, the cortex part is there in the primitive gonad. That is, the cortex part of the primitive gonad develops into an ovary. Then the primary female reproductive organ, that is, ovary is a primary female reproductive organ which is developed from the cortex of the primitive gonad. Okay? But the testes are developed from the medullary region of the primitive gonad. Conversely, the medullary region regresses or degenerates in females, and the embryonic ovary does not secrete any of the hormone at this stage. But in male, there are different type of hormones are secreted, some substances are secreted. But in case of female, there is no such steps are involved. There is no production of hormones at this stage. In girls also, Mullerian inhibitory substances are produced by granulosa cells in small follicles in the ovary, but plasma level remains very low or undetectable until puberty. Even girls also, the Mullerian inhibitory substances are produced. After puberty, the plasma Mullerian inhibitory substance levels are similar to the male. It's about 2 nanogram per ml. Then let's study the relationship between the androgen and the brain development. Okay. The development of the brain, which is particularly sexual behavior and control of gonadotropin secretion. Gonadotropins are the hormones that regulates the gonads, testes and the ovaries. Can be influenced by the hormone, that is androgens. Androgens, a class of male sex hormones. Okay. In certain animals such as rats, a brief exposure to the androgen during early days of development that leading to the development of male pattern of sexual behavior and the male pattern of hypothalamic control of gonadotropins, that secretion after puberty. Okay. If the animals are exposed to the androgen, that leads to the development of male sex characters. In other words, exposure to the androgen during the development can impact sexual behavior, but the hormonal control of the reproductive system remains its cyclic pattern. Means the cyclic pattern of gonadotropin secretion and other reproductive process such as menstrual cycle in females continue to function as expected. Okay, this suggested that certain aspects of sexual differentiation and reproductive function are resistant to the alteration by early exposure of the androgen. Okay, so sometimes some of the sexual behavior are controlled by the androgen hormones and some are not affected. The cyclic pattern remains as it is. Okay. Related to these hormonal exposure or hormonal secretion, there are some of the syndromes are observed in the females. For example, androgenital syndrome. That is androgenital syndrome. Androgenital syndrome, which results from the congenital adrenocortical enzyme deficiencies. That is congenital adrenocortical enzyme deficiency, which can lead to excess production of androgen. Women with this syndrome may exhibit masculized characters or the male characteristics, including behaviors due to the higher androgen level. I am repeating this. That is, the syndrome is called as androgenital syndrome. Androgenital syndrome is resulted from the congenital adrenocortical enzyme deficiency. Some of the enzyme which is released by the adrenocortical, that is congenital in nature, that is congenital adrenocortical enzyme deficiency. It can lead to the excess production of androgens, that is the male sex hormone in females, that result in the for production of masculinized characteristics or the male characteristics including behavior. Okay. This congenital adrenocortical enzyme deficiency is also known as congenital adrenal hyperplasia, that is CAH. Congenital adrenal hyperplasia. This is a group of inherited genetic disorders that affect the adrenal glands. Okay. This is the genetic disorder which affect the adrenal gland. So, congenital adrenal hyperplasia, CAH, or congenital adrenocortical enzyme deficiency is nogen. Okay. The adrenal gland produces mainly the three hormones, that is cortisol, aldosterone, and androgens. The most common form of congenital adrenal hyperplasia is due to a deficiency of an enzyme. That enzyme is called as 21 hydroxylase. The hydroxylase number 21. Without this enzyme, the adrenal glands are unable to produce sufficient amount of cortisol and aldosterone. So, the 21 hydroxylase enzyme deficiency lead to the deficiency of the enzyme cortisol and the aldosterone. So, resulting in an overproduction of androgen, that is male sex hormone. This can lead to a range of symptoms such as virilization. Virilization is the excess production of androgen that result in the development of male sexual characteristics, male secondary sexual characteristic. Okay. And another condition is the salt washing. Salt washing is the kind of electrolyte imbalance which is caused due to the deficiency of the hormone aldosterone. Okay. If the aldosterone level is very less, the individual is suffering from the salt wasting or the electrolyte imbalance. Then another condition is the hypoglycemia, that is low blood sugar level. Okay. This condition is resulted from the deficiency of the hormone cortisol. So, if the cortisol level is decreased, then the individual may experience hypoglycemia. It again a dangerous condition. Another one main important state is the irregular menstrual cycle and the infertility in female. Okay. Enzyme, that is 21 hydroxylase, is responsible for the condition called congenital adrenal hypoplasia or the congenital adrenocortical enzyme deficiency. This enzyme, that is 21 hydroxylase, is responsible for the deficiency is responsible for the overproduction of androgen by the deficiency of cortisol and the aldosterone. Okay. Then what are the treatment for this condition? That is the treatment for this condition is the hormone replacement therapy. Congenital adrenal hypoplasia may receive a cortisol replacement therapy to address the cortisol deficiency and reduce the overproduction of androgen. If we give cortisol to the patient with the condition that is congenital adrenogen hypoplasia, then androgen level we can limit. We can conclude that androgen, including testosterone, play a crucial role in the development of sexual behavior and the control of gonadotropin secretion. Understanding the impact of androgen on brain development and behavior is important for the comprehending the sexual differentiation in behavior in normal development. So, androgen or the testosterone level is very crucial for the control of brain and the production of sexual by the sexual hormones or the gonadotropins from the brain. Okay. Let's moving to the another important topic, that is aberrant sexual differentiation, that is abnormalities in the sexual differentiation. Okay, what are all the abnormalities we can see? First one is the chromosomal abnormalities in sexual development. Abnormalities in sexual development can be caused by various factors, including it may be a genetic or the hormonal irregularities or non-specific teratogenic influences. Okay, it may be a genetic, it may be hormonal irregularities or a non-specific teratogenic influences. What are these teratogens? Teratogenic influence, also known as teratogens, are the factor or the agent that cause the birth defects or abnormal development in the developing fetus during pregnancy. Teratogens are the agents that cause abnormalities in the fetus during the pregnancy itself. Okay. For example, if the mother takes some of the maternal drug such as alcohol, tobacco and some medication, that may causes the abnormalities in the fetus, or some if mother exposures to some of the infections like rubella or cytomegalovirus or Zika virus, can cause birth defects if contracted during the pregnancy. Or some other cases, that may be a radiation, that X-ray, radioactive material or chemicals, that lead, pesticides, insecticides, chemicals can lead to development of abnormalities. Okay. Some other cases is the poor nutrition status, that is folic acid deficiency if happens, that result in the neural tube defects and other developmental issue in the fetus. So here we discuss about the chromosomal abnormalities that play a significant role in sexual development. That is defects in the gametogenesis, that is gametes productions, eggs or sperm. Okay, the chromosomal abnormalities, whatever the abnormal chromosome or that chromosome which is result or develop into a defect in the gametogenesis, that chromosome linked to the gamete formation. If there is an abnormality occur in that chromosome, that may leads to the defected gametes or defected egg or defected sperm production. Okay. There are different type of chromosomal abnormalities we can see. First one is the non-disjunction of sex chromosome. Non-disjunction of sex chromosome. This is refers to the failure of sex chromosome to separate properly during the process of meiosis. Okay. Before formation of gametes, the cells are undergo meiotic division. During meiotic division, the chromosomes are separated. So, in this condition, that is non-disjunction of sex chromosome means here the sex chromosome should be separated during the meiotic division. But in this condition, there is a failure of sex chromosome to separate properly during the process of meiosis. This non-disjunction occurs during the first meiotic division, but it can also happen in the second meiotic division also, leading to the more complex chromosomal abnormalities. Okay. I will give you some syndrome or some pattern what resulted from this non-distinction of sex chromosome. One is the X0 condition, that is X0 chromosomal pattern. The syndrome is called as Turner syndrome. Non-distinction of one of the X chromosome during the oogenesis can result in the individual with the X0 chromosomal pattern, that is the absence of one X chromosome in female. In this condition, the individual have rudimentary or the absent gonads. No development of external genitalia. There is no genitalia in that individual. If the condition is X0 chromosomal pattern, Turner syndrome, there is an absence of external genitalia. And that individual having this syndrome showing short stature, that is individual's height is significantly below the average height. And some other congenital abnormalities are the birth defects and no sexual maturation at puberty. This condition refers to the gonadal dysgenesis, ovarian agenesis, or Turner syndrome. Other names for this syndrome, that is X0 pattern, chromosomal pattern, is known as Turner syndrome, gonadal dysgenesis, ovarian agenesis. Okay, here we can see there is absence of the X chromosome in female, result in the absence of external genitalia, and there is along with some birth defect also. Then another one non-distinction condition is XXY, that is chromosome pattern. It is known as Klinefelter syndrome. Okay. The next condition is the XXY, that is the presence of one extra Y. This XXY characterized pattern is most common in sex chromosomal disorder. Most common form of disorder is XXY. The individual with this pattern typically have the genitalia of the normal male. Okay. Here male genitalia is present. And testosterone secretion at puberty is also we observe, and development of male characteristic. But seminiferous tubules are abnormal. So there is a higher incidence of mental retardedness. We can see the mental retardedness in this individual with the XXY chromosome pattern or the Klinefelter syndrome. Okay. This condition is also known as seminiferous tubule dysgenesis or the Klinefelter syndrome. Here the male characters are developed and normal testosterone levels also we observe, but the individual is mentally retarded due to the seminiferous tubule is not in normal condition. Okay. The another pattern we observe is the XXX chromosome pattern or the triple X syndrome or the super female. We can see the super females. Okay. The XXX pattern or chromosomal pattern is relatively common and is often not associated with the characteristic abnormalities. Individuals with this pattern may not exhibit significant clinical feature, making it a less overt syndrome. Okay. There is a symptom clinically or external appearance are not differentiated with the normals. Okay. They are genetically having the pattern of XXX. Then another condition is Y0, where X in male is not present, and this condition is lethal. The another condition is the trisomy or the Down syndrome or Mongolism. This non-distinction of the chromosome number 21 leading to the trisomy 21. In this condition where there is an extra chromosome 21, this is Down syndrome is a pure example of gene excess causing abnormalities. That is, the presence of extra chromosome of number 21. So this extra chromosome that leads to the formation of Down syndrome. Okay, this is known as trisomy 21 or extra chromosome of number 21. How can we diagnose this condition? We can diagnose and prenatal screening also done during the pregnancy stage only or in fetus stage only. We can diagnose. Many chromosomal abnormalities and genetic diseases can be diagnosed during pregnancy through prenatal screening. Fetal cell obtained from amniotic fluid, that is, this process is known as amniocentesis, where the amniotic fluid is present in the outside the fetus. That amniotic fluid extracted, then screened for the abnormalities. Or chorionic villi or the chorionic villi sampling, that is by needle biopsy of this chorionic villi, we observe the villi can be analyzed for these chromosomal and genetic disorders or abnormalities. Okay, this much is about the chromosomal abnormalities. Let's moving to the hormonal abnormalities. As we all know, hormones play a critical role in the development of male and female genitalia during the fetal development. Okay. For example, pseudohermaphroditism is a condition in which an individual has the genetic constitution gonads of one sex but the external genitalia of the other sex. There are two types. One is female pseudohermaphroditism and male pseudohermaphroditism. Genetically, these individuals are one sex, but they have the external genitalia of other sex. In female pseudohermaphroditism, genetically female individual, the individual is genetically a female, but the individual has the male-like external genitalia. This condition resulted from the exposure of the androgen during the 8th and 13th week of gestation. It may be due to the congenital virilizing adrenal hyperplasia, where the adrenal gland produces the excess androgens. Okay. In male pseudohermaphroditism, where genetically the individual is male, but he is showing female-like external genitalia. This condition can occur due to the androgen resistant. Okay. In this condition, the tissue are less responsive to the male hormones, resulting in the incomplete masculinization. Okay. Androgen resistance can be caused by various mutations in the androgen receptor genes, leading to the range of defect in receptor functions. One form of androgen resistance is the 5-alpha reductase enzyme deficiency, where the enzyme responsible for the converting testosterone into dihydrotestosterone, which is the active form of testosterone. The 5-alpha enzyme reductase is the activation of the testosterone. Okay. If this enzyme deficiency occurs, the activation of testosterone is not taken place, so that there is an incomplete masculinization results and androgen resistance happen. This may be a case of mutation of the androgen receptor gene. Okay, this leads to the male pseudohermaphroditism. Then another one is the complete androgen resistance syndrome is there. That is result from the individual with male chromosome and normal testicular function, but external female genitalia of other cause the pseudohermaphroditism. This is the another condition where all the chromosome and testicular function are male are the normal, but the genitalia of the female. Okay, this is the another case. In some male, congenital blockage of pregnenolone formation. The pregnenolone, which is responsible for the production of testicular as well as the adrenal androgens. Okay, the androgen production, the male needs the pregnenolone. If the congenitally they are blocked, blockage in the pregnenolone formation, then this responsible for the release of androgen from the testicular as well as the adrenal. Okay. The congenital deficiency of the 17-alpha hydroxylase also responsible for the male hermaphroditism. Okay, na? The congenital deficiency of another enzyme, that is 17-alpha hydroxylase, also responsible for this condition. First one is 5-alpha reductase, and the one is the blockage of the pregnenolone formation, and the one is the 17-alpha hydroxylase enzyme deficiency. Okay. These are all the responsible for the hormonal abnormalities in the individuals. Okay. In both male and female, these hormonal abnormalities resulting in the pseudo-hermaphroditism. Then next moving to the another important topic, that is the puberty. What is puberty? What is puberty? Puberty is a natural and significant phase in human development when the body undergoes various changes and preparing the individual for the reproductive maturity. So, understanding of hormonal and physical changes that occurs during this period is essential for both medical and psychological well-being. Okay. Puberty is a critical phase in human development when the body undergoes significant changes, making the transition from the childhood to the adulthood. Is that clear? Then it is also characterized by the maturation of the reproductive system and the development of the secondary sexual characteristics. This process is largely characterized by the hormonal changes and typically occurs during the adolescence. Okay. In this puberty, many hormonal changes takes place, resulting in the reproductive system is developing and secondary sexual characters are also we can see. Okay. Puberty and the adolescence. Puberty, strictly defined, refers to the period when the endocrine and gametogenic function of the gonads, testes and ovaries have developed to the point where reproduction is possible. Okay. In the puberty stage, what happens? In this period, the endocrine as well as gametogenic function of the gonads, gonads are testes and the ovaries. Their functions, they show both the functions. That is, one is production of the endocrine hormones, that is sex hormones, as well as the production of the gametes, either sperm or the eggs. In the puberty stage, the hormones as well as the gametes are produced. Okay. So that in this stage, reproduction can be possible. Then some of the key events in the puberty. One is the thelarchy. Thelarchy is can observe in the girls. In girls, the first sign of puberty is called as thelarchy, which is development of the breast. Okay, thelarchy means the first sign of puberty, that is development of the breast. Then pubarchy. Pubarchy, this is the development of axillary or the underarm and pubic hair in both the sexes. Okay, pubarchy we can observe in the both the sexes, that is the development of the underarm and the pubic hairs. Then another one is the menarche. Menarche is observed only in the girls. That is, menarche is the first menstrual period in the girls. This first menstrual period, that is initial menstrual periods, are often anovulatory, that is lacking of ovulation, and regular ovulation typically occur after a year later. But the first sign of period is known as the menarche. Here ovulation is not occur. After 1 year of this first sign, then the ovulation begins. Okay. Then another one is the adrenarche. What is this adrenarche? Adrenarche is observed in the male, that is increase in the secretion of the adrenal glands. Adrenal gland start to produce androgens. Okay. Adrenarche is the increased production of androgens and that is result in the development of secondary sexual characteristics in the male. Then, hormonal changes during the puberty. Before or prior to the puberty, the gonads are inactive. But if they enter adolescence or the puberty, they are activated by the hormone called as gonadotropins. Gonadotropins are the hormones that activate the gonads to produce the sex hormones. Okay, gonadotropins are the gonad regulatory hormones from the pituitary glands. Okay, gonadotropins are the hormone which is released from the pituitary glands, and the function of this gonadotropins is the activation of the gonads. Okay, this initiates the final maturation of the reproductive system. Okay, before puberty, there is a slow release of estrogen and androgen secretion. We can observe in children between the age 7 and 10. Here, very slow release we can observe. When they enter to the pubertal stage, that is in teens, this slow rise of gonadal hormones proceeds the more rapid increase in their amount, which makes the onset of the puberty. Okay, initially the sex hormones such as estrogen and androgen levels are very less. Then its level increases in the teens, that is teenagers, which makes the onset of puberty. This hormonal synthesis and activation of gonads during puberty leading to the development of secondary sexual characters and ability to reproduce. The age at which individual enter to the puberty is variable and has experiences changed over time. Okay, in Europe and United States, it has shown trend of declining age over years. Okay, the typical age range between 8 to 13 years for girl, and the puberty age for male or boys is about 9 to 14 years. The adrenarche is the key event which involved in the increase of the secretion of adrenal androgens, which is primarily called as dehydroepiandrosterone. That is dehydroepiandrosterone, that is DHEA, which is typically occurs during the 8 to 10 years in the girls. In girls also, the slow level of the androgen release is taken place, and 10 to 12 years in boys. Okay. This dehydroepiandrosterone is an adrenal androgen that plays a crucial role in the development of the sexual characteristics. DHEA levels peaks at the age of 25 in females and slightly later in the male. DHEA is the precursor of the androgen, okay, primary form, after which they slowly decline to the low values in old ages. The level decreases or very low in the old ages. Okay, that is one of the important enzyme involved in the development of secondary sexual characters. And let's move into the factor influencing the timing of puberty. The timing of puberty is influenced by the genetic factors or the environmental factor as well as the nutritional factor. Changes in the nutrition, lifestyle and environmental condition have been linked to the observed decline in the age of puberty in recent days. If nutritional changes occur nowadays, so the age of puberty is decreased. During puberty, gonads are activated and secondary sexual characters were observed. This activation process is a complex and regulated by many processes. Here we can see several factors, hormones were involved in the regulation of this puberty. One such factor is the release of the gonadotropin releasing hormone. Okay, gonadotropins are required for the activation of the gonads. Okay, that hormone. For the gonadotropin's activation, the another hormone is required, that is gonadotropin releasing hormone, that is GnRH. Another factor is the body weight. The heavy body, fatty body are also influences the timing of puberty. Then let's study the role of gonadotropin releasing hormone in onset of puberty. As we all know, gonadotropins are hormones that stimulate the gonads. Okay, gonadotropin, which is secreted by the pituitary gland, which activates or stimulates the gonads, that is testes and the ovaries. The hypothalamus of brain releases this gonadotropin releasing hormone, which is in turn stimulates the pituitary gland to secrete the gonadotropins. Okay, hypothalamus releases the gonadotropin releasing hormone, which in turn stimulates the pituitary gland to secrete the gonadotropins. In pre-puberty children, gonadotropins are not produced, but there is a natural mechanism appear to inhibit the release of this gonadotropin releasing hormone. Okay, in small children, the gonadotropin releasing hormones are inactivated. Okay, they are not produced. One or more genes and their products involved in the stimulation of gonadotropin releasing hormones. One or more gene regulations is occur to activation of this process. Then another hormone is called as leptin, which is called as satiety producing hormone, which is secreted by the fat cell. May be linked between body weight and puberty. Okay. So studies in mice have shown that leptin is associated with the fertility, as obese mice lacking leptin are infertile, and fertility can be restored by injecting them with the leptins. Leptin treatment in female mice has been shown to induce precocious puberty. Leptin treatment in female mice have shown that induce the precocious puberty. Precocious puberty means the condition which is characterized by the early onset of sexual development and menstruation before the typical age range for puberty. Okay. Next important another condition is the brain lesions. Okay. Brain lesions and the puberty regulation. The regulation of the puberty is the complex process that involves various brain regions, including the hypothalamus and pituitary gland. As we all know, these two regions are very crucial because they synthesize or produces the puberty-related hormones. So, if any structural abnormalities occur in this region, there is an alteration in the puberty. For example, brain lesions. Brain lesions are the structural abnormalities or the damage in these critical areas that can interfere with the regulatory mechanism of puberty. Okay, the brain lesion interfere the neutral or the natural pathways responsible for the controlling of onset of puberty. Okay, the lesions or the tumors, or are there any other structural abnormalities in the brain region, that is in the hypothalamus as well as pituitary gland region, that result in the alteration of the puberty. The exact nature and the location of this lesion can impact how they affect the regulation of the puberty. This lesion may lead to an early release of gonadotropin, so releasing hormones. If the lesions may be responsible for this gonadotropin releasing hormone early release, then what happens, which in turn trigger the early activation of the gonads. Okay, this may be occurred. Then what are the causes for this brain lesion? Why these brain lesions are happen? This may be a congenital abnormalities by birth. Congenital abnormalities may occur. or any head injuries may be result in these abnormalities, or any tumors formation in the brain region may be responsible for these brain lesions. Okay, the presence of this brain lesion can lead to the medical condition known as the secondary central precociary puberty. That is, secondary central precociary puberty means the early onset of puberty. This is due to the brain lesions, that is damage to the central nervous system. This precociary puberty may be due to pineal tumors. Pineal tumors associated with the case of precociary puberty when they cause secondary damage to the hypothalamus region that disturb the normal regulation of the pulsatile gonadotropin releasing hormone secretion. If the individual have the pineal gland tumors, then what happens? The hypothalamus are affected. This what is the function of this hypothalamus? The hypothalamus secretes the gonadotropin releasing hormones. So if this condition, the hypothalamus is affected, so that gonadotropin releasing hormone secretion is also affected. Okay. Then what is the diagnosis and treatment for this precociary puberty? Diagnosis involves clinical assessment, brain imaging, and the hormonal level test. Okay. So these processes determines the presence of brain lesions and their impact on the puberty regulation. Treatment may include addressing the case of brain lesion, and in some cases hormone therapy to delay the onset of puberty. So hormone therapy, if we give, we can delay the puberty. So we conclude that the brain lesions can disturb the regulation of puberty, leading to the condition known as the precocious puberty or the early onset of puberty. So understanding the role of these brain lesions in this context is vital for diagnosis and appropriate medical investigation to ensure the well-being and health development of the affected individual. Then another condition is also there, that is delayed or the no puberty. There is no puberty. Some conditions are also there. This delayed puberty or the with no puberty is the absence or significant delay in the onset of secondary characteristics beyond the typical age range. Okay, if the delay occur in the puberty, but this condition, puberty, cannot be considered pathologically delayed until specific age markers are not reached. In some cases, we observe we have the age range for the onset of puberty, that is menarche, that is first menstruation, by the age of 17 for female, and testicular development by the age of 20. If the puberty is not occur in this age, then we can say that it is the delayed or the no puberty or absent of puberty. So this delayed puberty is maybe due to the endocrine abnormalities. So endocrine abnormality, hormonal abnormalities may result in the delayed puberty, such as panhypopituitarism, which is associated with the dwarfism and a range of endocrine issues. This is the endocrine abnormality, that is panhypopituitarism, where the dwarfism we can see, and also there is a high range of endocrine issues. Another condition is the X0 chromosomal pattern, or it is also known as the gonadal dysgenesis, also associated with the dwarfism. Okay. In some cases, despite of presence of gonads and normal endocrine function, delayed puberty occurs. In male, it's called as anicoidism, where there is a normal gonads and normal endocrine function, but also there is we can observe a delayed puberty. The condition of this in male is called as anicoidism, and also in female is known as amenorrhea, where we observe no puberty. Amenorrhea means no puberty. The reason is not known. Although the gonads and endocrine functions are all normal, but they have no puberty in female is known as amenorrhea. Okay. Primary amenorrhea, which is result in the gradual atrophy of the uterus and vagina due to reduced negative feedback effect of estrogen and progesterone on the secretion of follicle-stimulating hormone. Usually, primary amenorrhea occurs in the result of gradual decrease in the vagina as the uterus wall. So, this main reason for this condition is the negative feedback of the estrogen as well as progesterone. So, here follicle-stimulating hormones are affected. Premenopause and menopausal symptoms are seen in the individual who is entering to the menopause stage. Then what are these symptoms? Prior to the menopause, woman may experience a transition period called premenopause. Okay, prior to menopause, there is one transition period known as the premenopause, which can last up to 10 years, which is characterized by the irregular menstrual cycles and hormonal fluctuations. So, menopause symptoms includes hot flushes, that is warmth spreading from trunk to face, and the one is night sweats. These hot flushes coincide with the surge in luteinizing hormone secretion, but are not directly caused by luteinizing hormone, but the increased level of this luteinizing hormone may be responsible for the hot flush, that is heat of the body. These symptoms can last for many years and mainly impact on the woman's quality of life. Then what are the health implications? What happens for these changes? What makes the woman body? What changes are occur? In this menopause condition, the menopause is associated with an increased risk of various health conditions, including osteoporosis, this decreased bone density, and ischemic heart disease, and also renal or the kidney diseases. So, the decline in estrogen level, the estrogen levels are very decreased level, has a significant impact on the bone density and cardiovascular health. For all these health impact are resulted from the decreased level of the estrogen level. And then, I will ask you a question, whether andropause or the male menopause exists. Please answer me in the comment. Okay. What we study from this chapter, what we discussed, what we have studied, we studied about the intricate process of human reproductive system development, starting from the embryonic formation of gonads to the transformation phase of puberty and menopause. Okay. We also study the pivot role of sex chromosome, hormonal influences, and genetic abnormalities in shaping sexual differentiation. Main points, we observe that the differentiation of male and female reproductive organs depending on the presence of Y chromosome and specific endocrine systems such as testes in male and ovaries in female. At timings of puberty varies. Puberty onset is regulated by the secretion of the gonadotropin releasing hormone. Then leptin and body weight play a role in the timing of puberty. Variation in genetic or brain lesions including tumors, infection or hypothalamic abnormalities are responsible for the precocious and delayed puberty. Menopause marks the ends of a woman's reproductive years and is associated with the cessation of ovarian function. Overall, we discuss a panoramic view of human reproductive development, emphasizing the multifaceted factors that guide this remarkable journey. In next class, we will discuss about female and the male reproductive system, physiology, function and regulation. I refer to important books for these or all information, that is Guyton and Hall, Textbook of Medical Physiology, 12th edition, and Gannon's Review of Medical Physiology, 24th edition. So, if you like the video, please click like and share and subscribe our channel Bio Academy for the next video. Thank you.